Welcome, everyone. Thank you for joining today's webinar on fit-for-purpose water updates and lifestyle comparisons of non-potable water reuse scenarios. This webinar is part of the Water Research bi-monthly webinar series from EPA's Office of Research and Development. And we have a great presentation today. Before we get started, I'd just like to go over a few items. First is that there's a PDF copy of the slides located in the handouts of your GoToWebinar navigation panel. And that PDF will have all the links that we reference throughout this presentation. So be sure to use that if you're looking for any of those links. We'll be saving a few minutes at the end for a brief Q&A session. So we encourage you to submit questions throughout the duration of the presentation, and we'll get to as many as we can. So you can submit questions in the questions box that you should see in the GoToWebinar panel on your screen. If you experience any technical or audio issues, there's an email on the slide here, or you can type it in the chat box. And finally, the webinar will be recorded and will be posted to the Water Research Webinar Series webpage within 2 weeks of the webinar. And again, a link to that will be in the chat box on your GoToWebinar panel. And next slide, please. We'll give out certificates to those that requested it at registration. We just ask that we award them based on the requirements that you're in the room and registered, or you're in the room with someone who registered. You must attend the webinar for the full hour. And then finally, and this only applies if you have other people in the room with you, if those people want certificates, we need to know their names. So you can submit their names through the online form. And again, you can find that in your handout PDF or in the questions box in your panel. If you're watching the webinar at home alone, then you don't need to fill this form out. And then a few announcements on the next slide. A few upcoming webinars that we'd like to share. The next webinar in this series is on April 28th, and that will be on enhanced aquifer recharge. And there are 2 air-related webinars from EPA's Office of Research and Development upcoming in March. And on the next slide, we wanted to share an upcoming workshop. This workshop is accepting abstracts that just opened up, and the closing date for that window is April 20th. So if you'd like to present a technical talk, please do submit your abstract by then. You can find more information about that workshop at the link in the PDF handout. Next slide, please. So I'm pleased to introduce the presenters today. We have Jay Garland, Michael Janey, and Sissy Ma. Jay is a senior scientist with EPA's Office of Research and Development, Center for Environmental Solutions and Emergency Response. Michael is an environmental engineer, also with EPA's Center for Environmental Solutions and Emergency Response, or CESR, we call it. And Sissy Ma is a research engineer, also from EPA's CESR. And with that, I will pass it on to Jay Garland, who will kick off the presentation. Thank you again.
Thanks, Hannah. Can you hear me?
Yep, we can hear you.
Okay. First off, I want to apologize. I'm not on video because I'm having some technical difficulties. So I'm on the phone. But I'm glad to join you today. And I just want to give a brief kind of introduction to this topic before I turn over to Mike and Sissy to talk about some of the more details. So if you go to the next slide, I think that's slide 5. I think it's important. We use the term transformative approach that we're going to look at today in terms of this decentralized on-site non-potable reuse. And I think it's important to understand why are we worried about transforming the water sector. And I think it's important. The next couple of slides come from a recent paper by some real thought leaders in the area of water systems, both from the University of Michigan and Colorado State. They're listed there. And they point out that the main factors that really resulted in the current design of the systems we have no longer exist. And if you go advance to that next step in that slide, I mean, it talks about what those 2 factors are. I mean, when our current structures were defined, we had plenty of resources relative to availability, and we did not have the knowledge for treatment technologies and monitoring technologies. But we do now. We know how to do more advanced treatment, and we are facing more limited resource availability. So that really speaks to looking at water systems differently, thinking about it in a more integrated way, and thinking about it from a more resource recovery perspective. So if you go to the next slide, it kind of outlines a lot of the different factors as we transition the water sector and think about from historic to future, really thinking about water systems as part of the circular economy, producers of useful products, including energy and nutrients, thinking about more local water resources, and thinking about a continuum of decentralized and more centralized options, and really viewing it as a holistic approach, and really thinking about not just kind of plugging in the system into an already designed city, but really building the water, the new water systems into the planning for the city. So the next slide is kind of a very simple way of saying a lot of that. I mean, I think we think in the 21st century, wastewater infrastructure should safely produce resources. And in this case, I talk about materials and energy as the previous slide illustrated, but I also think that the recent experience that we've had with COVID-19 pandemic is that we've been using wastewater as a way to monitor the spread and prevalence of the disease. So wastewater we need to think about mining it, as that graphic illustrates, mining it for both the material resources, but the information resources in wastewater. So if you go to the next slide, we're going to zero in a little bit on this decentralized on-site non-potable reuse. And I think the way to view that from a resource recovery perspective is to think about buildings as producers of water. You can think about them as wastewater that's produced, but really turning it around and say, okay, buildings produce a variety of different waters, from roof collected rainwater to stormwater collected at grade to the actual combined wastewater flow out of the building to source separated graywater, even condensate from the AC system. So this variety of sources of water that could be tapped and reused locally. And if you go to the next slide, there's a number of increasing use of these examples. From the left slide, you see the Solar and Battery Park in New York City, which is one of the really early adopters of this kind of approach, where they've been processing wastewater on site through a membrane bioreactor and reusing it for cooling and irrigation and flushing toilets, a variety of non-potable uses for a couple decades now. And then moving towards the right, you see newer examples. The example in Hazel and 8th on Portland, it really thinks about trying to incorporate the treatment systems into desirable landscape elements. And then the final element is a recent example that just was commissioned and became operational in San Francisco. So if you go to the next slide, this slide kind of summarizes the work that the kind of the research effort that we developed in response to discussions with a group of people who were really interested in adopting safe on-site non-potable reuse. And that really is you see on the right-hand side, there's a number of different partners in here. The Blue Ribbon Commission for on-site non-potable reuse, that is really a group that we've been fortunate enough to work with from the inception, led by San Francisco Public Utility Commission, Paula Kehoe, but also involving utilities and public health agencies across the country who really wanted, like I said, to safely adopt and facilitate use of these kind of systems. But there was questions that came up, and really it's been a very great example of working with these end users, the stakeholders here, and really addressing key questions that they had to help facilitate that adoption. And we've really addressed three different questions. One is, how do you define acceptable treatment? If you're going to reuse these variety of different alternative waters for different end uses, how do you define that you're doing it safely? And that's the risk modeling that Michael's going to talk about in detail. The second element is that once you define the effect, how well the systems need to perform, how do you really monitor that they're performing as designed? That second element is something we continue to work on, and it is very important, because you can define the treatment standards, but really when it gets to implementation, you really need to know how to monitor the performance. And that's something that we've lagged on a little bit only because, I mentioned COVID earlier. The lead investigator on that, Nicole Brinkman, has really been dedicated almost full-time, or I'd say probably more than full-time, over the last year to the COVID wastewater issue. But that remains a core issue, but we're not going to give you any updates on that in this talk. And then finally, the question of, does this make sense to do this? And I think when we say that, we're thinking about from a holistic perspective. If you go to the next slide, it kind of describes when we say, does it make sense to do this? It means look at it holistically, really try to avoid burden shifting with respect to economic and environmental impacts. You can recycle that water locally, but make sure that you're thinking about the entirety of the system to make sure that you're not, like I said, just trading costs to somewhere else. And so Sissy is going to talk in detail about that. All right, so I'm going to turn it over to Mike now, who's going to get back to that first question of how do we define acceptable treatment.
Thanks,
Go ahead, Mike.
So how do you define acceptable treatment? These alternative source waters are unique from traditional water sources, and they arrive at the building untreated. And so there's a little bit of different of a management perspective on these. In addition, for decentralized wastewater collections, there's also scaling effects because there's limited dilution compared to municipal systems. And then finally, we're interested in this question of fit for purpose water. And so treating the different water sources only to the extent needed for their specific uses. And so we approach this by developing risk-based pathogen log reduction targets. Next slide, please. And we do this using a method called quantitative microbial risk assessment, or QMRA, which considers the exposure volume during different end uses, the density of pathogens in different source waters, and the respective dose response in order to predict risk for comparison against acceptable risk thresholds, and thereby defining what the treatment reduction should be. And we can do this for a matrix of different end uses and source waters, ranging from non-potable uses all the way to potable, and for different source waters of varying qualities. And by doing this, we can generate this set of fit for purpose reduction targets for different combinations of each source water and potential end use. Next slide, please. And so original set of LRTs was published in the report that you see here on the slide, with the key table featured on the right here. And so we developed log reduction targets for a variety of different source waters, domestic wastewater, graywater, stormwater, which we modeled two different qualities of stormwater, one with a low dilution of low contribution of municipal wastewater, and one with a high contribution of municipal wastewater, which I'll touch on again later, as well as roof runoff, which is what we call rainwater, just to distinguish the fact that it's collected above grade. The uses that we looked at were unrestricted irrigation. Sorry, can you go back? Thanks. The uses that we looked at were unrestricted irrigation, which means it doesn't need to be subsurface, but it does not include food crop irrigation. It's just for municipal uses. Indoor use, which is primarily for toilet flushing and clothes washing, but also includes a safety factor to protect against potential cross connections that could occur, or because these are on-site systems, there's potential for misuse of the different waters, and so we wanted to have some protection against that. And so the indoor use does include a rare accidental ingestion event affecting 10% of the population 1 day per year, which again I'll talk more about later. We've developed these LRTs for 3 different classes of pathogens: viruses, protozoa, and bacteria, with 2 different risk benchmarks. The main benchmark being 10 to the -4 infections per person per year, which is consistent with what's been used in the past for drinking water. In the parentheses, those numbers are for a more relaxed threshold of 10 to the -2 infections per person per year, which is more consistent with recreational exposures. And since this document has been out for a while, I'm going to focus today on the developments that have happened since its original publication. Next slide, please. And so the first area that we've been really looking at is the pathogenicity characterizations for the different water. And so for wastewater and graywater, again, these are decentralized collections. And so it requires a little bit different thinking. And so in these systems, pathogen infections can be intermittent because you just have a small number of people. And so just probabilistically, you won't always have pathogens present in that small community, whereas at the larger municipal levels, you're more likely to. There's also limited dilution effects. There's no stormwater influences, industrial inputs, other types of flows that would dilute out pathogen occurrences. And so we previously modeled this using an epidemiology-based simulation that considered the fecal contamination of different waters as well as the number of pathogens that are shed within a population of given sizes in order to model the pathogen concentrations in different types of wastewaters, including different types of gray water from different sources. We've since sought out to validate those measurements by looking at some decentralized systems. And so we've looked at one wastewater system and 2 graywater systems for 3 different viral pathogen targets, norovirus G1 and G2, as well as adenovirus, using 2 different molecular analysis methods, qPCR and droplet digital PCR, with the latter method selected because we really want to optimize our detections here. We know that sensitivity is going to be an issue. We're looking at some of these different water sources, and so by using qPCR, we're using digital PCR, and we're hoping to improve our sensitivity. Next slide, please. And so this graph compares our measurement results to the different percentiles of the simulation, which are the horizontal lines. And you can see that first that both qPCR and digital PCR agree well with each other, which is expected because they are both PCR-based methods, but it's also reassuring because we are counting on digital PCR to really get us to where we need to be for some of the other source waters. In terms of a comparison to the model, you can see that it really did reflect the median of the simulation extremely well, and also was able to characterize the variability. And so we were able to catch some of the high concentration events that really do end up driving your risk. I'll also say that we tried to look at seasonality, particularly for norovirus. It's a known seasonal infection. However, in the summer months on the right there, you can see that while the concentrations were on the low end of our detections, they were still within the range of what we measured throughout the rest of the year. So we had some perhaps limited effects of that in this case. And then finally, I'll point out that these concentrations are generally higher than what's reported in municipal wastewater collections. Those are average around 10 to the 4th per liter. Next slide, please. And so graywater, as I mentioned, it's a cleaner water source, and so we did expect to have fewer detections, which was the case. But we were able to quantify more samples using digital PCR than we were using qPCR. So that does point to the fact that that is an advantage of using the more sensitive analytical method. However, these detection rates were still lower than what was predicted by the model, and part of that is accounted for by the detection limits of the different methods, and actually a large quantity of the simulation results would be expected to be unmeasurable, even using our droplet digital PCR and high levels of sample concentration. And so that sort of suggests that there is still a role for modeling, even if you have measurements. You might not be able to measure all of the concentrations that are of interest from a public health standpoint. When we were able to measure concentrations in the few samples that they were quantifiable, they were within the range predicted by the simulations, but perhaps do not characterize that full range. So there is some value of additional data collection in this area. Next slide, please. For roof runoff, we previously modeled this based on animal fecal contamination. And so assuming that the fecal source in this case, rather than being human wastewater, is going to be animals that are using the roof. And so this was limited in our previous model to Gulf feces and bacterial pathogens, only because those are the only elements that we had sufficient data for. So certainly there are other animal species of interest, and there are other pathogens of interest as well. So we recognize that that's a limitation, and so we sought to try to better characterize the variety of pathogens that may be present using this off the roof study, which is a citizen science project. And again, in this case, we used droplet digital PCR and large volume samples, 10 liters, to try to maximize our detections, recognizing the detection limits were going to be an issue going into this. And so we collected samples, 80 samples across 4 cities and 13 different rain events. Next slide, please. And so our measured bacteria concentrations were within the model range. Our positive results were greater than the model median, but less than its maximum. And so we are right where we expect to be. And part of the reason that they will tend to be higher end measurements is because of the fact that we were unable to characterize low level measurements given our method limits of quantification. Again, we had lower detection than was predicted in the model. The model implicitly assumed that they were always going to be present, whereas in this case, they were not. And again, part of that is probably due to censoring of the measurements. We also measured protozoan pathogens in this study, which is particularly important because they're new, and there was our previous LRTs were specified only for bacteria because we had no data for protozoan pathogens, even though we suspected that they might be important. And so we have new detections that do reinforce that theory that rooftop collected rainwater should be treated for protozoan pathogens. But the problem here is that we only had 4 samples from 4 different sites that were positive, representing 5% of our total samples. And so even though this signals that an LRT is necessary for protozoan pathogens, the data is still fairly limited. Next slide, please. For stormwater, the challenge of stormwater is its variability. And so we took an approach to try to work around that by modeling it as different dilutions of municipal wastewater, with the reasoning being that municipal wastewater is going to be driving the pathogen risks in urban stormwater collections. And so this is valuable because it's a versatile and general approach, but it also creates a challenge because now we specified 2 different LRTs for different qualities of stormwater, and it results in some ambiguity for any project. They want to know, obviously, which LRT they should be using. And so within this area, our ongoing work is trying to develop new tools that can be used to better characterize where any site might fall with respect to these different dilutions of municipal wastewater. And so things like land cover, land use data, hydrologic modeling, sanitary surveys, information on the local asset conditions, and microbial source tracking markers, if you are going to take measurements, seem to be valuable approaches. Next slide, please. We've also looked at an additional source water that was not included in the original report. That's AC condensate, which is extremely high quality water that's generated in high quantities. And those quantities correlate with both water scarcity and also certain water demands. So cooling towers use a lot of water, and this is potentially a really good source for those applications, given its low TDS. But of course, the question is it safe? On the chemical side, because it is so pure, it can actually be corrosive. And so there's a concern about leaching of metals, which could particularly be important if you're going to use to irrigate plants. On the microbial side, we're really not anticipating any types of fecal inputs to condensate collections. And so the risk in this case really becomes opportunistic premise plumbing pathogens such as Legionella and Mycobacterium that are really an issue with any type of water management system, whether it's potable, reclaimed, or what have you. So we have started a pilot study to try to characterize the microbial quality of condensate collections. And we saw high HPC counts, which is a general indicator of microbial quality, as well as some detections of both Legionella and Mycobacterium in both the bulk water as well as biofilms within the piping. And so this does indicate that management for these types of premise plumbing pathogens should be considered. And we are working now to expand data collection through the Water Use Action Plan. Next slide, please. So shifting gears a little bit, another area that we're interested in is exposure assessment. And so the first thing that I'll talk about is the cross connections, which I mentioned previously. And so there's 2 different types of cross connections that are unique to non-potable water systems. The first being connections of reclaimed water to potable water supplies, and the second being a cross connection of the raw alternative water to non-potable water, which essentially equates to a treatment failure. And so what we wanted to know was whether our included ingestion safety factor was protective, and under what conditions it was. And so it turns out that for short duration, a small population and high dilution events, it is actually quite protective. And I'll further point out that when we looked at whether what the additional burden of including this cross connection was on the LRTs, we found that there was a less than 1 log increase in the LRTs when you include the cross connection event. And so to us, this signifies that there's a very good payoff for a small increase in LRT. Gives you a fairly robust amount of protection against periodic cross connection events, which are perhaps particularly important in on-site systems because there is no central management entity. Next slide, please. We're also interested in how sensitive our LRTs are to our exposure assumptions. These are admittedly uncertain. We're also interested in whether, given that uncertainty, if they can also be extended to other end uses with similar levels of exposure. So this graph here on the right shows the relationship between the log ingestion volume and the LRT for 2 different pathogens. In black is norovirus, and in gray is Campylobacter. And the solid lines on the plot show the scenarios that include that cross-connection event, a safety factor. And so you can see this plateau occurs where once you get to a small enough exposure volume, it is no longer sensitive to the exact volume. And so that means that if we are slightly off in some of our previous estimates, then that's okay as long as the safety factor is included. Likewise, it means that we can extend these same LRTs to other end uses that fall within that range of the plateau on the graph. And so this would include things like decorative fountains and vehicle washing. It would not include showering or bathing. And so we did develop a new set of LRTs for that use, which are available in the supplementary slides. Next slide, please. And as we were doing this, particularly as we started thinking about showering, we recognize that there's other pathogens besides enteric ones that we need to consider. And so for immersion exposures and different types of skin exposures, Staphylococcus aureus and methicillin-resistant Staph aureus, or MRSA, become important considerations. These can be high concentration in untreated source waters. However, this does require some additional considerations. We're talking about antimicrobial resistant pathogens. One of those being genetic exchange. And so if you're using biological treatment, are we providing opportunities to enhance the genetic transfer of these antibiotic resistance determinants? There's this concern for delayed infection following colonization. And so you may be colonized by a resistant strain, have no problems until you develop an infection from it later down the line, and you have to go to the hospital to get treated, and you can't get treated for it because it's a resistant strain. And then thereby that can cause all kinds of problems in terms of your overall outcome. And again, along the same lines, clinical severity of illness. If you have an infection that's untreatable, clearly you're going to have increased morbidity and mortality associated with that particular type of infection. And so in this case, we took the similar approach to some of our previous analyses to say whether our LRTs are protective against our enteric-based LRTs are protective against these types of pathogens as well. And it appears that they might result in low infection risks, but again, that doesn't really capture the complete outcome. And so previously, our endpoint of our analysis was infections, and then we compared it to the 10 to the minus 4 infections per person per year benchmark. In this case, we don't think that's sufficient because you really want to be able to characterize the severity of those infections. And so we're doing that using a disability adjusted life year approach, which has a benchmark of 10 to the minus 6 per person per year. Next slide, please. And then finally, we also wanted to evaluate whether systems that are complying with indicator-based standards also achieve acceptable levels of public health risk. And so we looked at the systems that are improved under the NSF 350 standard and then ran the risk model to see what the risk associated with those systems is for different types of waters and different scales of application. And so the first system is an MBR, to which we added chlorine disinfection, recognizing that any state regulator is going to require that as well. And the second is a septic tank with a sand filter and UV disinfection. For the latter case, we did not have any data, and so I'm just going to focus on the first case, for which the manufacturer provides treatment data with respect to viral removals. And so we actually see, depending on the different source waters, they can result in a varying level of risk, ranging from low risk for small-scale graywater systems to medium risk for business collections to quite high risk for wastewater and certain graywater collections. And this result was not surprising because obviously they did not specifically consider the associated risk when they developed these standards. And even more importantly, it doesn't consider viral or protozoan pathogens. These are based on fecal indicator bacteria, whereas the other pathogen classes are really driving the risk that you see here. And so we're not considering them in development of the system, or at least approval of the system. Then we would not expect them to necessarily achieve that. And that is what we see. So this really does emphasize that you need to consider, the risk-based approaches are necessary in order to get to where you need to be. Next slide, please. And so in conclusion, risk-based treatment targets are designed to remove enteric pathogens to protect human health, but LRTs are sensitive to input water type intended use in accidental ingestion. improve pathogen characterization, support the previous LRTs, but they also provide data to update them. The built-in safety factor is protected for short-term, low-magnitude cross-connection events, and existing LRTs can be extended to other uses with small exposure volumes under certain conditions. Additional considerations are necessary to incorporate skin pathogens and antimicrobial resistance into QMRA. Fecal indicator-based treatment systems have a variable predicted level of public health protection. And finally, we developed new RTs for bathing and showering, as well as intermediate-sized systems that are available in the supplementary slides. And with that, I'll pass it on to Sissy. Thanks.
Okay, so now we understand how safe is safe for non-potable water reuse. The next question is, does it make sense, and how do we design those systems? So in the last couple of years, 7 years, we have been explored the NPR systems using system metrics like life cycle assessment to explore different parameters in those configuration. And the latest one that we're working on is the large urban buildings, like in San Francisco, and using membrane bioreactor to do different source water treatment, like a mixed wastewater or source separated graywater. And we expand that efforts in developing a tool for the NPR applications. So using a life cycle approach to evaluate non-potable water reuse, basically the idea is to capture the life cycle, upstream and downstream, upstream supply chain and downstream emission of this whole process. Eventually, you have a water that you use it from centralized drinking water supply, and then after you use it, the wastewater, the sludge is just being discharged to the centralized wastewater. But in the process, the mixed wastewater or the graywater can be reused. So this is a circling system that goes through this the red boxes, these unit processes. The green box, the green arrow here, these are the ones the energy can be offset, or the water can be offset by this non-potable water use process. And the unit process in the middle, the aerobic NBR, anaerobic NBR, and recirculating vertical flow wetlands are the main unit process that we explore and we're comparing here. So this is a transitional system. The sludge is still being treated to the centralized system, so it's not completely separate, independent. And these emissions is being considered too. So these are the unit process, the treatment train that we are considering. Aerobic NBRs, anaerobic NBRs, and the wetland system. And you can see it's pretty typical pre-treatment, biological treatment, post-treatment, and also disinfection. But because of the different log reduction target that each of the unit process can bring, so the design of the treatment train will be different. For example, the wetland system, and it's not as effective to remove the pathogens. So the additional disinfection of the ozone unit process is being added. So you can, these are the information you will see later, you'll reflect it in the results. So here is an example of the results, the global warming potential. So here, these are the scenarios in unit process that we are exploring. The blue one is the aerobic NBRs with the thermal recovery from the hot water heater. And the red one is the one without the thermal recovery. And the green one is the anaerobic one. The purple one is anaerobic NBR with the intermittent sparging treatment, and the yellow one was the wetlands. So you can see this gray block, the bars are the one that's drinking water that you're offsetting, you're avoiding by using non-potable on-site water reuse. So the overall, the net value is the aerobic NBR perform better because it recovers quite a bit of energy from the hot water heater. And also another difference is this is the graywater versus the wastewater, the mixed wastewater. So graywater perform a little bit better than the wastewater because it's cleaner. So this is the cost side, and you can see actually the aerobic still perform better than the rest of the scenario. But in this case, wastewater perform a little bit better in terms of the cost than the graywater because graywater require a separate additional plumbing system, the piping system. So for this integrated assessment study that we found out, the display of the drinking water is a real benefit for the on-site water reuse. And the important thing is to match the scale of the treatment system. You want to maximize the potable water that you can offset, but you don't want to over-treat it. Or you can find additional venues, additional revenue. If you, as a building owner, you can, sell that water to your neighbors or to the city. The mixed wastewater aerobic NBR is the most economical treatment options. The anaerobic NBR and the wetland, because they are emerging technology and the data that we find in the literature is more variable, so that affects the cost and the impacts. So maybe when the technology become more mature, things might change, and the graywater system, because of additional costs and that adds to the total cost for the graywater system. The important thing for the thermal recovery is actually turns out it's quite a bit of an impact to the overall system. So that's how much energy that you can offset in your region. So when we study with the San Francisco scenarios in that climate, and we realized in a different city or different geographic location or different climate, the outcome might be different. So we expand those knowledge and develop a tool to help different utilities or building owners in a different location try to explore these options. So this is a tool that we actually just launched yesterday. So now you can go to this website, explore it. So basically, you go to this website, you can access through this button here. The question is, if you want to do water reuse, and what's the most economic and environmentally most effective way to do that, and what's the source water that's available to meet the large urban building non-potable water needs. So the audience for this tool, intended audience, building developer, building owners that they want to explore this option, and the urban communities that in urban planner and want to explore this option too. And certifications institution like Green Building Council, of course, scientists wants to explore more non-potable water options. So basically, when you go to this site, it's zip code based. So you go in and plug in your zip code, the building different characteristics, what type of building, if it's a mixed use, is it commercial or residential, and how many floors, and what's the square footage, occupant for the building, and what's the source water? Do you have rainwater, AC condensate, or you just want to consider wastewater? Do you want to consider thermal recovery, and what's the hot water heater that you have? And do you have a separate piping for separate graywater, or you intended to? And for your appliance, is it high efficiency or not? And the end use, what you're going to use the water for, is it laundry, is it toilet flushing, or irrigation? So these are options that you can explore. So results like this will be for example, the results zip code based. So you can see throughout the year how much of water, the rainwater that you're going to have, or the potential of the AC condensate that you could explore. So here, the next result is water availability and the demand. So the dots and the line is the water demand. So how much water that you needed for the non-potable use. And you can see for this zip code, the mixed wastewater is much more greater than the demand. And of course, in the summertime, there's more rain and also the AC condensate that adds into that the supply side. So we explored throughout the country, the zip code, different zip code, different area, what's the potential for rain harvesting and AC condensate. And you can see majority of the country, the rain potential, most of the country does not have much rainfall. The northwest and southeast has more potential. Similar on the AC condensate side, northeast had more potential. So relative to the rain harvesting and AC condensate, most of the regions less than 10% to satisfy the water demand. Here, only part of the Florida has more than 20%. But in all cases, wastewater and graywater can satisfy all demands, the non-potable demands. So we want to explore further the geographic location and the building characteristics and how that affects the performance. So have 3 scenarios. And the first one, we explore the different location with a 40,000 zip code that's explored. So the second scenario, we want to explore the cost, the drinking water price cost. What is that coming to play? So the third one is the consider all the variabilities, the geographic location and building characteristics. So what we found, for example, here is the global warming potential. The green dots here are the AC condensate, the blue is the rain harvesting potential, and gray water is the brown one, and the yellow one is the wastewater. So you can see this bigger picture. When the volume, when the annual water can be delivered, when the quantity is small, you actually take more energy per unit of water to produce that. So it's the global warming potential is high, but there's a certain window here, 50,000 to 500,000 gallons per year. This is the region that doing rainwater harvesting or AC condensate, it's good benefit. But when the system gets larger, the wastewater, the NBRs come into play, and it'll be better. And on the right side here, explore the effects of the thermal recovery in terms of different egret region. You can see when you recover natural gas consumption, and this is the effect, and to recover the electricity based on hot water heater, especially in the egret region that's more high carbon grid, you get more benefits. So again, here is a more visualized thermal recovery. On the top 2 panel is the offsetting the natural gas. The bottom 2 is an offsetting the electricity. The natural gas one here, you can see New York here on the northeast and on the west side. These are the relatively cleaner egret region. The electricity is cleaner. So when they off the natural gas, because of the natural gas use a heat pump, so you can actually offset better in these regions. For the electricity, offsetting the electricity hot water heater, the high carbon grid, for example, in the Midwest, will see the most benefits. So in terms of the cost, on the left side, you can see the economy of scale come into play. Obviously, when you go into the larger quantity of water, wastewater, the NBRs are more beneficial. And similar here, when you compare it to the drinking water costs, when you want to offset the water cost and the higher water cost, you see more benefits. So the geographic analysis, we found out that rainwater and AC condensate really can only provide less than 10% of the non-potable water needs for the majority of the country. So when it's available, these sources can provide more environmental benefit but it's costly to do that. On the other hand, wastewater and graywater can play 100% of the demand, but it has a lot to do with energy use for the treatment system. So it's really dependent on where you locate in the region and how clean your electricity grid is. And so the local drinking water costs, drinking water that you try to offset play a big role too. So this is a tool that we can provide so the users can explore the local specification, the regional specification, design what you need and what's the cost benefits. So the next step with this tool is released. There's more things that we can do. For example, the rainwater and AC condensate, there's an option you can combine them. You can combine the storage, the tank, that can bring down the cost. And how about the stormwater, the foundation drainage, and some users mentioned the cooling water. So these are the ones that we want to explore. And of course, you can expand this and usage, the analysis to the community scale. Right now, it's only the building scale. So also, there's an indirect impact. So you divert a lot of water from your sewer system. And what is the potential reduction for CSOs and eutrophication, also cost to your wastewater treatment plant. So there's some ongoing and potential collaboration that we were exploring using these NEWR tool with the San Francisco and Cincinnati here, and supporting some efforts, the non-potable water use efforts in New York.
Thank you, Jay, Michael, and Sissy for your fantastic presentations, and thank you everyone for joining. And that's it for today. Join us in 2 months for a webinar on enhanced aquifer recharge. Thanks very much.